morning, my dear students. Welcome to CCLMS Media Center, Visakhapatnam. I am Dr. V. Padmaja, working as lecturer in Botany in Dr. V.S. Krishna Government Degree College, Visakhapatnam. Today, I will discuss a topic that is sterilization, which was included in the third semester paper Plant Biotechnology in Botany major course. Okay, now we will discuss about sterilization. Sterilization is one of the important aspects in plant tissue culture. I think you all know about plant tissue culture you have studied in previous classes. What is plant tissue culture? Plant tissue culture is the culture of plant cells or tissues or organs on artificial nutrient medium under aseptic conditions. Through plant tissue culture, we can generate more number of plants in less space. That is why it is called micropropagation. There are some steps in plant tissue culture. To do the plant tissue culture technique, first we have to prepare the nutrient medium. Then we have to sterilize the nutrient medium. Then we have to prepare the explant and sterilize the explant. The next step is inoculation of the sterilized explant onto the sterilized nutrient medium. This must be again done under aseptic or sterilized conditions. Then the inoculated culture vessels are incubated in the incubation chamber or the incubation room. After the development of the plantlets, they must be hardened and they must be acclimatized to the outer environment. This is briefly the technique of plant tissue culture. So, sterilization is very important to do the plant tissue culture technique. The entire plant tissue culture lab must be in sterilized condition. Not only the lab, the persons who are doing the tissue culture technique, they also must wear the sterilized dresses. The culture vials must be sterilized, the nutrient medium must be sterilized, the explants must be sterilized, incubation room must be sterilized, and all the things must be in sterilized condition for the plant tissue culture technique. Then only the technique will be successful. So, we know from this discussion, I already discussed that sterilization is very important or one of the important aspects of plant tissue culture technique. Now, I will discuss about sterilization. What is sterilization? Now, I will discuss what is sterilization. Sterilization means it is a process of elimination of microorganisms. So, while doing tissue culture technique, we have to eliminate microorganisms. Otherwise, what happens is the microorganisms will infect the plant tissues and the tissue culture will be failed. Therefore, sterilization is the elimination of microorganisms, and it is very important to eliminate microorganisms in the tissue culture technique. So, the maintenance of aseptic conditions or sterilized conditions is highly essential for successful plant tissue culture technique. In vitro plant tissue culture, often microorganisms like bacteria or viruses or fungi infect the cultures. Then the tissue culture will be failed. Therefore, it is essential to eliminate the microorganisms from the tissue culture. Therefore, all the things which are used in the tissue culture must be maintained in aseptic condition. We use nutrient media, explants, inoculating instruments, culture tubes, culture vials, culture rooms, incubation rooms, etc. All these rooms, inoculating instruments, etc. must be maintained in completely sterilized condition. Then only the culture will be successful. Now, I will discuss about methods of sterilization. There are mainly two methods of sterilization. First one is physical methods of sterilization. Second one is chemical methods of sterilization. Now, I will discuss about physical methods of sterilization. Physical methods of sterilization are again classified into three categories: sterilization by heat, sterilization by radiation, and filter sterilization. Now I will discuss about sterilization by heat. The name itself indicates that here the physical factor heat is used to sterilize the tissue culture instruments. It is the most simplest and the reliable method of sterilization. Here heat is applied for sterilization technique. Again heat is applied in two forms, dry heat and wet heat. Now I will discuss about dry heat sterilization. Generally, this dry heat sterilization is used for sterilizing the glassware or glass instruments like petri plates or test tubes, pipettes and other inoculation instruments. This dry heat is provided in an instrument called hot air oven. In this hot air oven, generally the glass vials or glass instruments are sterilized at 160 to 170 degrees centigrade for 1 to 2 hours. Then all forms of microorganisms will be eliminated. Therefore, so we have to sterilize the instruments which are used in the tissue culture through hot air oven by applying the dry heat. This is the photograph of hot air oven. Here there will be an adjustment or regulation for temperature and time also. We have to keep the glass instruments in an aluminum foil and kept in the hot air oven. Then the glass instruments will be sterilized in this instrument. Next one is wet heat or moist heat sterilization. The name itself indicates that heat is applied in moist state. That means heat is applied in the form of steam under pressure. We observe this in our pressure cookers also. In moist heat sterilization, moist heat in the form of steam is applied under pressure. It provides tremendous heat above the boiling point. Using this moist heat also, we can do the sterilization. This moist heat is provided in an apparatus called autoclave. This instrument is used for sterilizing the nutrient medium. In autoclave also there will be regulation for temperature and pressure. Generally, some of the nutrient medium components are sterilized in this autoclave. This is the photograph of the autoclave. It works just like a pressure cooker. In this instrument, moist heat under pressure is provided and nutrient medium is kept inside this instrument and it will be sterilized. The standard conditions for autoclaving are 121 degrees centigrade at 15 psi pressure. Psi means pascals per square inch pressure must be provided for 20 minutes. Under these conditions, the nutrient media components will be sterilized. The sterilized medium only must be used in the tissue culture technique. If without sterilization the nutrient medium is used, then there will be an infection of microorganisms and failure of entire tissue culture process. Advantages of autoclave are speed, simplicity and destruction of viruses. There are some disadvantages of using this instrument also. There will be a change in pH by 0.3 to 0.5 units. Change in pH is not accepted in plant tissue culture technique because the correct pH only gives the right result, and there will be separation of nutrient medium components or sometimes undesirable chemical reactions also occur in the nutrient medium. These are some of the disadvantages of using the autoclave. Second method which comes under physical methods is sterilization by radiation or radiation sterilization. There are many forms of radiations, but in tissue culture technique, ultraviolet radiation is used for sterilization. This UV light generally eliminates different microorganisms. UV light is used, radiation in the form of UV light is used to sterilize inoculation chambers and culture rooms. The disposable plasticwares we use are also UV sterilized. The sterilized autoclaved medium is then transferred into sterilized containers in the laminar airflow cabinet. In the laminar airflow cabinet, radiation in the form of UV light is provided. This radiation is useful for sterilization during inoculation. Inoculation means transfer of sterilized explant onto the nutrient medium, which is also sterilized. The transfer of sterilized explant onto the sterilized nutrient medium must also be done under aseptic or sterilized conditions. So, laminar airflow chamber is used for this purpose. In laminar airflow chamber, complete aseptic environment is provided through providing UV light in the form of UV lamps, etc. This is the photograph of laminar airflow chamber. Inside this glass chamber, inoculation is done. Inoculation means transfer of sterilized explant onto the sterilized nutrient medium by using some sterilized instruments. Next, third type of sterilization which comes under physical methods is filter sterilization. Generally, some of the components which are used in the nutrient media cannot tolerate heat. Therefore, they cannot be sterilized by using an autoclave. Therefore, they must be subjected to filter sterilization. Filtration is used to remove microorganisms from solutions which are heat sensitive. Some components of the nutrient medium like enzymes, phytohormones, etc. cannot be autoclaved because they are heat sensitive. Therefore, they must be subjected to filtration for the eradication of the microorganisms. Different types of filters are used to remove the microorganisms like bacterial endospores, fungal spores, etc. These filters which are used in filter sterilization are made from different materials like asbestos, diatomaceous earth or biological membranes or cellulose papers. So, generally in filter sterilization, membrane filters and millipore filters are most frequently used. These are mostly used for sterilization. Inoculation chambers and culture rooms are also filter sterilized. Here, filter sterilization is used to filter the air from the microbes because, as you all know, microbes are abundantly present in the air. So, during inoculation, there may be a chance of infection of the plant tissues present in the air. Therefore, inoculation, that is the transfer of sterilized explant onto the sterilized nutrient medium, must be done under aseptic conditions. I already discussed that this is done under laminar airflow chamber. In laminar airflow chamber, microbial free air is provided by HEPA filters, that is high efficiency particulate air filters. These HEPA air filters, they filter the microbes from the air and provide the microbial free air in the inoculation chamber or laminar airflow chamber. So, these are the three methods of physical sterilization I have discussed: sterilization by heat, sterilization by radiation and sterilization by filters or filter sterilization. Now, I will discuss the second method of sterilization, that is chemical methods of sterilization. The name itself indicates that here we use chemicals for the sterilization procedure. Generally, explants are sterilized by using different types of chemicals. I already discussed that explants must be in sterilized state. What is an explant? Explant is any part of the plant which is used to culture in plant tissue culture technique. As it is excised from the plant and the plant is in the outer environment, number of microbes will be seen on the explant. Therefore, it is highly essential to sterilize the explant also. We use different types of explants in tissue culture like root tips, shoot tips, leaves, anthers, pollen, ovule, ovary, embryo, seeds, etc. Any part of the plant, or even we take single cells also for plant tissue culture. Sometimes protoplasts also will be taken. Any part of the plant can be used as explant for plant tissue culture. As they are excised from the plants and microbes may be present on these explants, we have to sterilize the explants. So, for sterilizing the explants, we use different types of chemicals. These chemicals may be again surface sterilants, disinfectants, antibiotics, etc. So, what is chemical method of sterilization? It is the eradication of microorganisms with the help of chemicals like surface sterilants, disinfectants, antibiotics, etc. Surface sterilants are the chemicals which remove the microbes present on the surface of the explant. Disinfectants are generally used for treating the explant, and they will eradicate the microbes present in the explant. Antibiotics, you all know, they will eradicate the bacteria, etc. So, explants used in plant tissue culture like seeds, tissue pieces, any tissue piece can be used for plant tissue culture like tissue pieces, excised embryos, pollen grains, anthers, etc. are treated with disinfectants. There are different types of disinfectants like hydrogen peroxide, sodium hypochlorite, mercury chloride, etc. We can use any type of chemical for sterilization of the explant. Generally, seeds are surface sterilized by treating them with mercury chloride. There is some specificity of chemicals. Some parts of the plant or some explants are surface sterilized with specific chemicals. Like that, seeds are generally sterilized by using the chemical mercury chloride. For other parts of the plant, mostly sodium hypochlorite is used for sterilization. After treating with chemicals, we have to wash the explant with distilled water 3 or 4 times so that the chemical will be removed from that explant. Okay, so after sterilization, after chemical sterilization, we have to compulsorily wash the explant with distilled water 3 to 4 times. So, there must be a provision for distillation water plant in the tissue culture room also. We have to use only distilled water in the tissue culture technique. Chemicals, we also generally use alcohol also for chemical sterilization. Most of the inoculation instruments are sterilized by dipping in alcohol followed by flame sterilization. Alcohol is also one of the chemicals used for sterilizing the explants. After alcohol, as you all know, generally 70% alcohol is used for sterilization. Inoculation instruments like forceps, needles, spatulas, all the inoculation instruments are sterilized by dipping them in 70% alcohol, and then they must be flame sterilized also. Then they will be free from microbes. Then only we have to use those inoculating instruments in the plant tissue culture technique. Some of the common surface sterilants or some of the common chemicals which are used to sterilize the explants I have given here. 1% sodium hypochlorite solution. Mostly this chemical is used to sterilize most of the explants in plant tissue culture technique. 4 to 10 grams per 100 ml of calcium hypochlorite. This chemical can also be used. 1% solution of bromine water. 0.01 to 1% solution of mercury chloride. 70% alcohol. I already told you that inoculation instruments are first sterilized by dipping them in alcohol followed by flame sterilization. 10% hydrogen peroxide, 1% silver nitrate, 4 to 50 milligrams per liter of antibiotic solution. We can also add antibiotics to eradicate the microorganisms like bacteria, fungi, etc. Even though we take care of sterilization, the bacterial endospores and the fungal spores, they can resist all the conditions and they can survive in the nutrient medium. Therefore, in some cases we have to add antibiotics also to the nutrient medium. In this way, we can successfully do the plant tissue culture technique and generate the plants what we desire. After sterilization only, successful sterilization only, we have to sterilize the nutrient medium, we have to sterilize the explants, we have to sterilize the inoculating instruments, we have to sterilize the culture rooms, we have to sterilize the incubation rooms, etc. Then only the plant tissue culture will be successful. Therefore, sterilization is very important aspect in the plant tissue culture. Thank you. Thank you one and all.